Let's go, Stefan.
This we will manage. Okay.
Is this okay? Is my hair okay? Should I change something? No?
No, you are perfect as you are, Alex. Welcome to Easy Immunology. My name is Stefan, and if you have not seen one of our videos, we try to explain you the relatively complex immune system in rather easy terms. And today we want to have a high-level overview over the complete immune system, and I never do this alone, as you might know. I have again recruited two very talented, two very knowledgeable ladies. Both hold at least a master's degree in biology, and they will support me with questions, asking questions that you might have so that we can directly clarify them within the video. And I have again invited with me Alex. Hi, Alex.
Hi, Stefan.
And Hannah. Hi, Hannah.
Hi, Stefan.
So, as I said, we will do a general overview over the complete immune system in one video, and then in the following videos we will go into much more detail into all the different parts of the immune system. Before we start, I want to throw in a little disclaimer. This is George Box. He is a rather famous statistician, mathematician. And he said a very useful sentence. He said that essentially all models, he meant of course in mathematics, are wrong, but some are useful. And the same is true for our look at the immune system. So I will try to explain this rather complex and not in all details understood immune system to you in very easy terms. At least I try. But as you can imagine, everything is much more complicated. And as I said, many things are also not understood in complete detail. So please keep this in mind as we go forward. So the function of our immune system is to protect us from pathogens. Pathogens are organisms or viruses that want to make you sick. And there are quite a different array of pathogens. And I always start this kind of lecture also when I give it to students by asking the students, in this case Alex and Hannah, if they know different kinds of pathogens. And also maybe you watching the video can press pause and think about it for a few seconds and then continue with the video. So what kind of pathogens do we have, ladies? Yeah, Alex.
I just start with fungi.
Fungi, yeah. What else do we have? Hannah?
Bacteria and viruses.
Okay.
Parasites.
Parasites. Are there different parasites, Alex?
And there are also prions.
Prions, yeah. And there are different kinds of parasites.
The multi-cell parasites and the one-cell parasite.
Yeah, the single-cell parasites like malaria and the big nasty parasites, the worms that cause us quite a little bit of trouble. So here are the different pathogens, and you have to keep in mind that you are always surrounded by these pathogens and your immune system is constantly fighting them. But most of the time you do not feel sick, so our immune system is very good at eliminating most of them, and today we try to get a high-level understanding on how this works. That these pathogens are very different is, I think, quite obvious. But here I just grouped them according to one parameter, which is size. And you can see that prions, which are misfolded proteins, are very small. Then viruses are quite a bit bigger, like 10 times, 15 times bigger. They are aggregates of dozens or 100 proteins. Then we have bacteria, which are real single-celled organisms. Then we have the single-cell parasites, which are real eukaryotes. They have a nucleus and are much more complicated. Their genome is like 10, 15, 50 times bigger than the typical genome of a bacterium. Then we have fungi, which are as big as our cells in our body. And then we have the multicellular parasites that start at a few millimeters. You can see them with your bare eyes. And then we have like real big nasty parasites that can be up to 12 meters long. If they live, for example, in our gut. So you see, there's a huge, vast difference already in terms of size of these pathogens. And therefore, you can probably very easily understand that the immune system needs to develop quite different mechanisms to cope with these different pathogens. And this is what we want to look at today. So there are many different ways to group the immune system, and I have decided to group it into 4 different components. The first ones are the anatomical barriers. They exist everywhere where you potentially have contact with the outside. So you have to imagine the outside of your body is usually non-sterile. It's full of these pathogens. And your inside is usually sterile, so there are no pathogens inside under ideal circumstances. And these anatomical barriers prevent, by certain mechanisms, that the pathogens come from the non-sterile outside into the sterile inside of your body. You find them on the skin, the mucosa in your mouth, and your epithelium in your gut, in your lungs, in your reproductive tract, everywhere where you potentially have contact with the outside world. So if you look at these barriers in more detail, we start with the anatomical barriers, and the first part of the anatomical barriers are the so-called mechanical barriers, and they are formed by epithelial cells that we find in our skin, our gut, our lungs, and in the nose and the eye. So what are these cells? They are specialized cells. They look like this, and they are basically a 1-cell thick layer that separates the non-sterile outside with potentially a lot of pathogens from the sterile inside of your body. And what now happens if you get infected is that either these pathogens destroy these epithelial cells and thereby invade your body, or the pathogens just get into your body because you have, for example, a wound that has nothing to do with an infection. Maybe you fell from your bike, you have a wound and you get some bacteria inside, and then this epithelial barrier, that's how we call it, is disrupted and the pathogens get inside your body. The epithelial layer is, as I said, only 1-cell layer thick, and this is pretty cool. It keeps out a lot of pathogens, but many pathogens are very, very small. Like bacteria can be very small and viruses can be even smaller, and they can squeeze through the area between the different cells, and therefore these cells have developed mechanisms to stick together even better. And this is done by so-called tight junction proteins. They stick together very, very strongly and basically prevent anything from coming through this barrier. And you can see here on the right side an electron microscopical picture. In white you see the nucleus of the epithelial cells. In red you see the cytoplasm, so it's the liquid inside the cell, if you want to call it like this. And in green you see these tight junction proteins, and you see that they form a very tight seal around these epithelial cells. Alex, you have a question?
When we look at the epithelial barrier of the skin, when we scratch our skin, there's something beneath. But this isn't epithelial cells, right?
No, beneath you have the keratinocytes and the first immune cells, like the Langerhans cells, dendritic cells that patrol the skin, and this is a problem. So there are some diseases like allergies where people start to scratch, or infections when people start to scratch, and then you destroy this barrier and you bring basically the pathogens via scratching more into the inside of your body, and this causes a lot of problems.
So if I have an allergy and the skin is attached, so it's easier to get infected with other bacteria also.
Yes, that's true. And also if you have a bacterial infection and the epithelium is damaged, it's easier to get infected with viruses or fungi or something, because this epithelial barrier is disrupted and a part of your immune system is not functioning anymore.
Thank you.
Okay. In addition to the mechanical barriers formed by the epithelial cells with the tight junctions, we have other types of mechanical barriers. You have, for example, the longitudinal flow of air and fluid in the skin and in the gut. So what does this mean? So basically your skin, your gut, especially your gut and your lungs, are inside your body, but they're basically connected directly to the outside. So there we have all the pathogens that are normally outside in your body because you eat non-sterile food and you breathe in non-sterile air. And by just moving like I do when giving this lecture, I just shake off some bacteria. This is on the skin and also in your gut. There's some movement because of the muscles and the fluid in your gut, and this just washes away certain bacteria. The same you have in the lungs. So the lungs produce something that is called mucus. It's basically glibbery stuff. You notice once you have had a cold, you produce a lot of mucus and it comes out of your nose and out of your lung. And this mucus is transported towards your mouth and your nose via cilia. These are small hairs that just push the mucus out, and with the mucus, a lot of pathogens are also pushed out. And your eyes and your nose produce tears. You notice this is fluid. There's some proteins also in there that are not so healthy for pathogens. And also in your nose, you have hairs that push out, for example, bacteria if you have a cold. Then we have chemical barriers. So they are based on chemical molecules, chemical reactions. For example, on all barriers, you have so-called antimicrobial peptides, which damage pathogens. We will look at this in more detail in a second. In the skin, you have fatty acids. So these are special molecules that integrate into the membrane of, for example, bacteria, and they disturb the function of that membrane. So this is not so healthy. In the gut, you probably know you have a very low pH in your stomach and in your gut, and you have enzymes that degrade proteins like pepsin. So this is not very welcoming. In the lungs, we have something you have probably never heard of. It's called pulmonary surfactant. It's molecules that are both lipophilic and non-lipophilic. So these are molecules that have parts that are soluble in water and parts that are not soluble in water, and they just reduce the surface tension in the lung, in this mucus, for example. And this allows bacteria not to colonize your mucus, but they sink in the mucus towards the bottom of the lung, where the epithelial cells are, and then special immune cells directly take them up and destroy them. And in the tears, you have enzymes like lysozymes that degrade certain bacterial cell walls. So if you have small children, they cannot clean their nose if they're very young. You give them lysozyme tablets that they can take, and this is like pure enzyme that destroys bacteria, and this helps them control infections. And then you have the microbiological barrier. So this just means that none of the outer contact points of your body with your surrounding is totally sterile, but it's colonized with certain bacteria. So you have a microbiome on your skin, in your lung, in your gut, in your reproductive tract, and these are usually bacteria that you live together happily, and they just colonize these niches and prevent the bad guys and girls from colonizing your lung and your gut. So this is just competition via some good microorganisms. So, as I said, these anatomical barriers prevent usually the entry of the pathogen from the non-sterile outside into the sterile inside of your body. But some pathogens have evolved mechanism to just destroy these barriers, to pass them, or if you have an infection, a wound, then these pathogens can just go through that wound, for example. And once they find themselves in the sterile inside of your body, they can start causing damage. And to prevent this, we have additional layers of protection. And the second component layer of protection is the complement system and the antimicrobial proteins. So the complement system is quite complicated. Remember my disclaimer. So Alex and me once tried to understand the complement system, and we went through 50 pages in the most famous textbook of immunology, and I think at least I came out more confused in the end than what I went in. So I will try to make it very, very simple. So imagine you are a bacterium and you have invaded Stefan's body, and you have certain structures on your surface, like these black pili. on this blue bacterium. And my immune system have made antibodies because I have had, for example, contact with a similar bacterium before. So my immune system knows what a black pili is and that it belongs to a bacterium. And these antibodies are everywhere in my body, and they now directly bind to the surface of that bacterium and trigger the so-called complement system. And this is a cascade of proteins that bind to each other and activate each other. And it starts with a complement protein 1 called C1 binding to antibodies. This recruits then 2 additional complement proteins, complement protein 2 and 4. And together, if they come together with C1, they are cleaved into big fragments and smaller fragments. They are totally confusingly named differently. Here the bigger fragment is called C2A and the smaller fragment is C2B, and for C4 it's the other way around. So the bigger fragment is called C4B and the smaller one is called C4A. I'm sorry. I didn't invent it, it's just how it is. But basically C4B and C2A then group together and form something that is called a C3 convertase, because it binds the C3 molecule and converts it again into a big fragment called C3b and a small fragment called C3a. And together, these effects have certain consequences. The first one is that the bacterium gets marked on the outside with all these complement proteins. They stick like a pimple outside of the bacterium. And also the bacterium swims in a cloud of these small molecules which do not stick to the surface of the bacterium, but they surround the bacterium. And cells of our immune system, like phagocytes, like macrophages, we will get to them in a second. They know if they find high concentrations of these small complement proteins, something is off. Therefore, they follow the concentration from lower concentrations to higher concentrations, and thereby they find the place where the complement cascade was activated and therefore where the bacterium is located. And then they bind with certain receptors to the complement proteins that are fixed on the surface of the bacterium, and then they can either kill it by mechanisms that we will look at in a few slides, or just phagocytose it, so they eat up the bacterium and then it's destroyed. And this bringing the phagocytes to the place of infection is called recruitment. This is via the small soluble fragments. And the marking of the bacterium as dangerous and foreign is called opsonization, and this is done by the fragments that stick to the bacterium. So this is cool. This eliminates a lot of pathogens already, but the complement cascade is not done yet, because this C3b together with C4b and C2a forms something that is called the C5 convertase that cleaves then the complement protein 5, and then it goes on and on and on. And basically at the end of the complement cascade, a protein that is called C9 is integrated into the membrane. And this is a pore-forming protein. So we poke small holes into the outer shell of the bacterium, and then the inner parts of the bacterium will flow out uncontrollably, and this leads to the death of the bacterium. So this is called cytolysis. Lysis is to dissolve something, and cyto is the cells. So the cells are dissolved because they basically bleed out. And the cool thing is that this is something that we call a complement cascade. So it starts with very few antibodies and very few molecules of C1, but they then activate more molecules of C2 and C4. They then activate even more molecules of C3, so it's multiplying. So we get a very strong response and a very efficient elimination of pathogens by the complement system. So the complement system has basically 3 important steps. The first one is the opsonization, where the pathogen gets marked as dangerous and foreign by either the binding of antibodies or complement proteins. Then we have the induction of an immune response by recruiting and activating different cells of the immune system. And this results in inflammation. So an activation of the immune system results in inflammation. We will later on see what are the characteristics of an inflammation, and this results in also damage in your body. So therefore, that's why I colored it red, because this will lead to you not feeling so well, because in the process of destroying the bacterium, for example, also parts of your body can be damaged. And then there is the phagocytosis and killing of the invading pathogen that then results in us being protected from said pathogen. There are different ways to activate the complement system. We will do this very fast. You can press on pause and compare the 3 cartoons on the right side, and you will see that, for example, between the first and the second cartoon, the only difference is that the antibody is missing. So what I explained you before is something that we call the classical pathway, where antibodies result in the activation of the complement system. So they trigger the complement cascade in the beginning. Then it obviously also works without antibodies, and this is called the alternative pathway. And this works because the most important protein in the complement cascade is the C3 protein, and this is unstable in a normal solution in your body, so it will just spontaneously be degraded into a big C3b and a small C3a fragment. And the big fragment then can just, if it is around or near a pathogen, can stick to the surface of the pathogen, and this will lead to the complement cascade being triggered. It's not as efficient as if it's triggered by an antibody, but it still works. And your cells in your body, at least most of them, have evolved mechanisms to directly remove the C3 again, so that they do not get damaged by this mechanism. Then finally, we have the so-called lectin pathway. There, a special protein, it's a mannose binding lectin. It's a protein that has specialized in recognizing certain sugar structures that mostly only exist on bacteria and fungi, replaces the antibody in initially triggering the complement cascade. In addition to the complement cascade, we have the so-called antimicrobial proteins or antimicrobial peptides. Here you see a red bacterium that comes into contact after invading our body with these antimicrobial peptides. And these are small proteins, so they consist of amino acids, and the amino acid sequence is structured in that way that we have a very highly polar structure. So one part of the protein is very positively charged because there are amino acids that have a positive charge because of their side chain. And then there is a part that is not so positively charged. And these proteins, at least that is the mechanism that it is postulated at the moment, integrate into the membrane of the red bacterium, and then the positively charged parts start to repel each other, and therefore they just push holes, they rip holes into the surface of the bacterium, and then again the inside of the bacterium flows out uncontrollably, and it's basically a more simple way of cytolysis that is induced by these antimicrobial peptides. And these antimicrobial peptides are everywhere in your body, so they are a very efficient but very unspecific way of killing certain pathogens. This is the second barrier, so the complement system and the antimicrobial proteins eliminate also many pathogens directly after they have invaded your body. As I said, they get a lot of the bad guys and girls, but not all of them. And for the rest, we need the innate and the adaptive immune system. And these are now specialized cells that are a bit more complicated, I would say, for the innate immune system, and much more complicated for the adaptive immune system. So we will start with the innate immune system. So, as I said, this consists of cells, and first we want to look at the overall spectrum of cells in our immune system. And the immune system is separated into two parts. The first one is the innate immune system, It consists of, for example, dendritic cells and macrophages, cells that eat up pathogens. Also neutrophils can do this. Then we have eosinophils, mast cells, and basophils. We also have NK cells and NKT cells, so quite an array of different cells. And they develop, all the cells of the immune system develop in so-called primary lymphoid organs. So we will look at this in much more detail in later lectures, but basically you have, if you are a grown up, you have two primary lymphoid organs. You have the bone marrow and you have the thymus. And in the bone marrow, we have so-called pluripotent stem cells. So these are cells that have decided they want to be something. They can turn into many different cells in your body, not all of them. So it's not an omnipotent stem cell, like an oocyte would be an omnipotent stem cell that could develop into your whole body. It has to, basically, because this is what you start at. But these pluripotent stem cells are one layer of differentiation further, so they can turn into many different things, but not into all. And this then further develops into a common lymphoid progenitor that will form these cells of the adaptive immune system, the T and the B cells, and the common myeloid progenitor. And this forms now all the cells of the innate immune system, like the macrophage, the neutrophil, the dendritic cells, and all the eosinophils, basophils, and mast cells. So that's how it works. This all happens either completely in the bone marrow, and the cells leave the bone marrow as fully trained, fully differentiated immune cells, or for example, for mast cells, they leave the bone marrow as progenitor cells, so as not fully trained cells, and they then develop in the periphery, in the parts of your body. Immunologically, the periphery is everything that is not the bone marrow or the thymus, and then they then turn into fully trained immune cells in the periphery. In the bone marrow, also the common lymphoid progenitor develops into a B cell progenitor that then gives rise to a B cell. And the common lymphoid progenitor also wanders from the bone marrow to the thymus, fascinatingly, and there turns into a T cell progenitor that then develops into a T cell. These are very complex processes that are not completely understood. So I think for T and B cells, we have a quite good grasp, and we will explain you this in the following lectures. But for example, here for the common lymphoid progenitor, it's not completely clear what happens when, everywhere, and there are different steps in between. It's quite complicated. But I think if you start trying to understand the immune system, I think this is already complicated enough. So the cells of the innate immune system have one major task, and this is distinguishing self, so all the cells in my or Alex or Hannah's body, from everything that is non-self. So these are all these pathogens, but this is also your lunch. So this is your steak, your salad, your vegan sausage, whatever you like. So this is also non-self usually, but this is usually not dangerous unless you are allergic to it. And the immune system has to find out, okay, what is self? So what is components of my own body and tissue and cells, and what is non-self? And for the non-self, it has to distinguish, okay, is this something Stefan just ate that he needs to thrive and to give this lecture, or is this something that is dangerous that our immune system has to fight? And therefore, the immune system has to sort out what is foreign, and among the foreign things also what is dangerous. And this is done by cells that we call antigen-presenting cells. So they are the first line of sorting out all the things that come into your body, and they make basically the decision whether an immune response should be initiated or not. So the antigen-presenting cell is a complex beast that is also not completely understood, but I will try to make it quite simple. So imagine this blue bacterium has again managed to invade our body, to make it past the complement system and the antimicrobial proteins. And now it meets its end. It meets an antigen-presenting cell. And these pathogens have certain structures that we call pumps, pathogen-associated molecular patterns. And these are just molecules that only exist in these pathogens, but not in our own cells and tissues. And I've brought you one example here. This is the bacterial flagella. So the flagella is a structure, it's a whip-like structure that is like a screwdriver turning itself around, and by this it generates motion that propels the bacterium forward in your body and also outside of your body. And this is a structure that consists of one single protein. It's called flagellin, and this only exists in bacteria, because we also have flagella-like structures, cilia in your nose, remember, from getting the mucus from your lung through your nose, but they look totally different. They look much more complicated. There are dozens of different proteins involved. So the flagellin only exists in these bacteria. And therefore, if a flagellin is around, this means a bacterium is around, and this is potentially dangerous. And therefore, our immune system has developed a structure that specifically recognizes this flagellin, and this is called a pattern recognition receptor. That is a receptor you can imagine like a key that fits into a keyhole. So it's a structure that very specifically recognizes flagellin. And then the immune cell gets activated. So, as I said, there are different pathogen-associated molecular patterns. They are, for example, this flagellum can also be cell wall components or nucleic acids inside of bacteria or viruses. And they bind to these pattern recognition receptors, and they trigger then immune cell activation. This is a very important concept in immunology that was also rewarded with the Nobel Prize in 2011 to Beutler, Hoffman, and Steinman. The cells of the immune system, of the innate immune system, have many different of these pattern recognition receptors. Here you see a dendritic cell and a macrophage. These are the two main types of antigen-presenting cells. And you see there are quite many different types. They also look very different. Pattern recognition receptors, both on the surface and on the inside of the cell. And I put you some examples here. It's not so important for you, but there are, for example, toll-like receptors that recognize flagellin, for example, toll-like receptor 5. Then you have toll-like receptor 4 that recognizes certain sugar lipid structures called lipopolysaccharides that are typically for gram-negative bacteria. There are intracellular receptors, so it's quite diverse. The most famous family of pattern recognition receptors are the so-called toll-like receptors. The toll-like receptors are called toll-like in abbreviation marks because they were discovered in a fly, the fruit fly, Drosophila. It's a very commonly used model organism in genetics. And these flies can be irradiated, resulting in random mutations in genes in the fly. And then you can generate lines from these flies, and depending on where the mutation is, these flies, for example, have no antennas, or they have no wings, or lack certain structures in their bodies. And in one of these lines, also the toll gene was destroyed, and this resulted in a defect in the immune system of the fly, because also flies have an immune system. And they couldn't fight fungi. And these flies were covered with nets of fungi. It looked very, very funky. And the postdoc that did the experiment then showed this to his boss, Miss Professor Nüsslein-Vollhardt, very famous geneticist, also Nobel Prize winner. And she just said, "Oh, toll." She was German, and toll means great. And therefore, these genes are called toll genes in Drosophila. And some years later, we also found the respective counterparts in invertebrates, in humans, in mice, and rats. And they are called toll-like receptors. That's where the toll comes from. And there are basically two groups of toll-like receptors. The first group are the receptors that are located on the outside of the cell. And they recognize structures on the outside of the pathogen. So remember, one hand is the immune cell and the other hand is the pathogen. Only structures that are outside of the pathogen can be recognized also by receptors that are outside on the immune cell. Then we have a second group of toll-like receptors 3, 7, and 9. They are located in the endosome. So this is in the inside of the immune cell in a special vesicle that's called the endosome. And there things end up that the cell takes up from the outside, and also certain processes inside the cell result in proteins that end up in the endosome. And if a cell is infected, for example, by certain viruses, they try to replicate in, for example, the endosome or in the cytoplasm. And these parts or these amino acids of the viruses and bacteria are then recognized as dangerous and foreign by the toll-like receptors in the endosome. For example, double-stranded RNA is something that doesn't exist in our replication cycle, but it exists in the replication cycle of certain viruses, and therefore the presence of large amounts of double-stranded RNA is a sign that the cell has been compromised and infected, and it needs to start a reaction to fight that pathogen. As I said, the toll-like receptors are the most famous group of pattern recognition receptors, but there are more. There are C-type lectin receptors that recognize certain sugar structures that we find on bacteria and also fungi that are very important in fighting these pathogens. They're also on the outside because these sugar structures are also on the outside of the pathogens. And then we have 3 more types of receptors that we find on the inside. We have the knot-like receptor in the cytoplasm, so in the liquid filling of the cell that recognizes certain structures from pathogens. For example, I told you flagellin is recognized via toll-like receptor 5, and this is again an example for where I try to make things very simple, because flagellin, the part of the flagella, is recognized by toll-like receptor 5 if it's outside of the cell. If it is inside of the cell, it is recognized by a knot-like receptor, and therefore the cell can even distinguish, is the flagellin outside? Then the bacterium is still outside, or is the flagellin inside? This means I have already been infected as an immune cell. I need to start maybe a more stronger immune response. So quite fascinating. Then we have the Rig-like receptors that recognize viral RNA, and we have the cytosolic DNA sensor that recognizes microbial DNA. So if now such an antigen-presenting cell recognizes a pump via its pattern recognition receptors, it transmits an activating signal into the nucleus of that cell, and this results in gene transcription. So there are nice models where you can infect these antigen-presenting cells with viruses, and of the 22,000 different genes in our genome, up to 8,000 are differentially regulated in a virus-infected cells. So they turn on and turn off a lot of different genes. So they start making a lot of different stuff. For example, they express certain co-stimulatory molecules on their surface. So these are molecules that are only there if this process has happened, if the cell has found something dangerous. So it's basically a translation of the information that something dangerous has been found by the antigen-presenting cell into the expression of these molecules. They also make cytokines, very soluble small molecules that transmit information between cells in our body. And they always take up things from their surrounding, but if their pattern recognition receptors are activated, this is even enhanced. And they take up, for example, the flagellin or the whole bacterium in their inside into so-called endolysosomes. These fuse with lysosomes. These are vesicles filled with proteases, and therefore the proteins, the flagellin, gets degraded into small fragments that we call peptides, like 8 to 12 amino acids in length. And these peptides are then presented on the surface of the antigen-presenting cell in complex with so-called major histocompatibility complex proteins. That's why the cell is called antigen-presenting cell, because it shows on, it's outside what it's working on in the inside. And if this happens, the cell moves to the lymph node, a structure that we will learn in future lectures. And there it provides 3 signals to a naive T cell. So a naive T cell is a T cell that has never seen a pathogen and it doesn't know if it's useful or not, because as we will learn later on, these receptors that the T cell uses to recognize pathogens, the T cell receptor, that's the green and gray part, are generated randomly. And in the beginning, they don't know if this is even useful. But if the T cell receptor is able to recognize the peptide presented by the MHC complex, this is called signal 1, it knows, okay, the antigen presenting cell is showing me something that was taken up by her, so it's part of a pathogen probably. Then it gets stuck to the cell, and this results in first interaction, but it's not sticky enough, not long enough to really fully activate the cell. Therefore, we need the signal 2. These are the co-stimulatory molecules that provide additional signaling through the nucleus to activate the T cell. And this is usually enough to fully activate a T cell, but then it usually doesn't know what to do because it just sees the peptide. It has no eyes. It does not know what a bacterium is, and therefore it needs an additional information. This is transmitted via the cytokines. They are called signal 3, and they then are transmitting information whether the cell should turn into an antibacterial T cell, antiviral, and antifungal or something T cell. So for the activation of T cells, we always need 3 signals. The first one, peptide MHC to T cell receptor, makes sure that the correct T cell is activated. Signal 2, co-stimulatory molecules, make sure that this only happens once the cell has seen a pathogen and its pattern recognition receptors have been activated. And signal 3, the cytokines, make sure that T cell knows what to do and what kind of immune response it needs to start. So once this happens, the T cell gets activated. It turns from a naive T cell that has never seen its antigen into something that we call an effector T cell. You see it, it looks a bit different. It's a bit bigger. It has decided to be an antibacterial T cell, if you want to call it like this. And these T cells are in this case called T helper cells, and they now orchestrate the immune response toward the pathogen. This is what we want to look at in the remainder of the lecture. And the main way how these effector T cells, helper T cells, orchestrate the immune response are called cytokines. So I always tell my students the effector T cells are like the managers of the immune system. They do not do the actual work. They just tell other people what to do, and they tell other people what to do by these cytokines. And they produce large amounts of different combinations of cytokines depending on what kind of immune response should be initiated. And cytokines can, for example, result in the activation of phagocytes. Phagocytes, like this macrophage shown here, are cells that specialize in eating up pathogens. So imagine this is a phagocyte that meets a bacterium. Then the bacterium is recognized by certain receptors, pattern recognition receptors. Then the phagocyte flows around that pathogen until it finds itself in the inside of the phagocyte in something that we call an endosome again. And the endosome then fuses with lysosomes, which contain a lot of proteases and toxic molecules. And then the bacterium gets degraded within these endolysosomes into peptides, and then the bacterium is gone. That's what phagocytes do. And then they usually also get activated in this process because they realize, oh, I'm eating up a lot of bacteria. Maybe I should call for help because maybe I'm alone. I can eat up 3, 4, 5 dozen bacteria, but I cannot kill all of them. And therefore they also produce cytokines and chemokines, messenger molecules of the immune system that attract even more cells of the immune system to the place of infection. And together, this activation of phagocytes The most famous type are the so-called macrophages, results in an inflammation. So an activation of the immune system is usually paralleled by a process that we call inflammation. And I've brought an example here. This is a hand, obviously, and you can see that the whole hand is inflamed. It's quite red, and it was also itchy, and it hurt. And it's a good example of an inflammation, but I want to make it a little bit more easy to understand by breaking it down into the so-called 5 cardinal signs of inflammation. So imagine this is our small scientist, and he has an infection with a bacterium in his finger. And this results in the activation of the phagocytes by the bacterium. They eat up the pathogens. They secrete cytokines and chemokines, and they induce an inflammation. And then our little researcher will develop the 5 cardinal signs of inflammation. So first one is that he will feel heat, maybe only in the finger in the beginning, but if he develops fever, then he will feel the heat in his whole body. Then it will turn red, the infected or inflamed tissue. Then he will feel pain because things are damaged during that infection. Then it can swell. So you see the hand is quite a bit bigger because there's swelling going on. And if this goes on for a longer time, he will not be able to use his hand. So if you have a good, strong inflammation, you will not be able to use your finger. So this is what we call loss of function. And these 5 signs of inflammation were discovered nearly 2,000 years ago by Celze and Galen. One was a Greek scientist, and the other one was a Italian or Roman scientist. And therefore these signs of inflammation have Roman names. So heat is called calor, redness is called rubor, pain is called dolor, swelling is a tumor, and loss of function is called functiolese. So these are the typical signs of inflammation that you can also use in your day-to-day life to judge if something is inflamed. And I think then it's quite obvious. So let's now look where these signs of inflammation come from. So imagine you have your bacterium that has managed to infect your body, and the upper cell layer is your skin, and above that layer is outside, and below that layer is inside. Then phagocytes will eat up the pathogen, produce cytokines and chemokines, and these cytokines and chemokines bind to their target cells in all of your body, but they do so in a concentration-dependent manner. So the amount concentration of cytokines and chemokines will be the highest around the area of infection, because that's where they're produced, and then they diffuse away, and therefore you find the highest amounts of cytokines and chemokines at the place of infection. They also change the properties of the cells in your blood vessels. You see the blood vessel is small, and the cells in the blood vessel wall stick together very closely. Here the blood vessel has expanded and has also become more permissive for cells to come in and out, and this is what happens. So if your blood vessels increase in diameter, the tissue will turn red because there's more blood flowing through the vessels, and therefore it will be a redder color. And also, if more fluid is pumped through the tissue, then it would also turn warm. So therefore we have heat and redness already. Then the other cells of the immune system will be recruited, will be directed to the place of infections by these cytokines and chemokines. So they have receptors for these cytokines and chemokines, and they wander from the place of low concentration towards the higher concentration of these cytokines and chemokines. And by this, they find the place of infection, and then in the vicinity of the place of infection, they leave the blood vessel, migrate into the tissue, and start forming an immune response to kill the remaining pathogens. And since we have more fluid that also evades the blood vessels and more cells in the tissue, it will turn bigger, there's more volume, it will swell. And since activation of the immune system results maybe also in damage towards structures in your body, this can also result in pain. And if this goes on for a longer time, we will have a loss of function in there. So this is how the 5 signs of inflammation are induced. Yes, Alex?
Is there only one way for the cells to arrive at the inflammation site?
No, there are multiple ways. So I told you that the cells wander there via the blood vessels. That is one way. And cells need like 24 hours to pass every spot in your body via the blood vessels. But there's also the lymphatic system. This is a parallel system where mostly the immune cells are wandering around. And you have, in some tissues, you have also immune cells always in the tissue. So they are there waiting for the immune response to start. So there are specialized dendritic cells, for example, like Langerhans cells in the skin. There are special macrophages in the liver. They're called Kupfer cells. They are special Frankenstein cells between dendritic cells and macrophages in the brain that are called microglia cells. So there are different ways. It's not like the tissue is totally empty and devoid of immune cells, but there's always somebody around because it can be a matter of minutes to eliminate the pathogens before they cause too much damage.
And maybe I just attached to this answer, is there also places in your body where are none or only few immune cells?
Yes, there are certain places in your body. They are called immunoprivileged sites because there are not so many immune cells around, because as you can imagine, the immune system is like a broad sword that can cause a lot of damage. And some structures in your body have the tendency to not regrow after an immune response, like your eyes, like your brain, like your testes. So therefore, immune cells are kept out of certain parts in your body in order to not cause too much damage. But if these spaces in your body are infected, then immune cells will come in there. For example, in the brain, I think that is the best understood. We have the blood-brain barrier. These are specialized cells that control what goes in and out of the brain in order to avoid unnecessary damage to the brain.
Okay, thank you.
Okay. So these are the phagocytes that cause inflammation. Also, cytokines produced by T cells can induce the activation of neutrophils. Neutrophils are fascinating cells. Alex loves them. I also love them because they are like the Swiss Army knife of the immune system. They are very important. Like 80% of the immune cells in the blood in humans are these neutrophils. And they can do, broadly speaking, 4 different things. So they can do phagocytosis. They can take up pathogens and eat them. They can produce so-called reactive oxygen species. So these are molecules that are very reactive because you see this fat point on these molecules. I will highlight it in post. They have something that we call a free electron. So electrons are parts of the atoms in your body. And electrons always want to be in pairs. They never want to be alone. And these molecules are generated by the neutrophils, and they have single electrons. And this is what we call free radicals. They are very, very keen on finding a second electron again, and they just rip out electrons from the next molecule that they find, and thereby damage these molecules. And these reactive oxygen species are produced in large amounts in something that we call an oxidative burst, because this results in these cells using a lot of oxygen to generate these reactive oxygen species. And then the bacteria find themselves in a cloud of these reactive oxygen species, and they rip out electrons left and right from all kinds of molecules from the bacteria, and therefore they damage the bacteria. Then they can also degranulate, so they have granules, vesicles in their inside they can just release. And there are, for example, hydrolytic enzymes that cleave molecular bonds between between atoms. They have myeloperoxidase, lysozyme, lactoferrin. These are enzymes that either destroy the cell wall of bacteria, they take away iron molecules that are important for enzymes in these bacteria, and they have alkaline phosphatase that also damages molecules, and the myeloperoxidase produces even more reactive oxygen species. And then I think that's the coolest part is they can degrade their nucleus and change the structure of their DNA and shoot these DNA sticky molecules out like nets, like Spider-Man, and trap these pathogens and keep them away from your body. And this is a process that is called net formation or netosis, because they form these neutrophil extracellular traps, nets. I think it's a nice abbreviation. So very important cells that have very diverse functions, but they are like one use cells. So if they undergo this reactive oxygen process or net formation, they are certainly dead afterwards. Degranulation and phagocytosis can decap that for a bit more longer, but they are like one-use items, and therefore we have so many of them because they always get damaged in the process. If these cells do not work, I brought you some clinical examples. So here on the left side, you have granuloma. You see these small structures, and these are basically patients that have a defect in the neutrophils, and the immune system starts to fight pathogens, for example in the skin here or under the skin, and it realizes, okay, there's a pathogen, I want to start an immune response, but I cannot fight them because my immune system is not functioning correctly. And at some point the immune system just gives up and it puts the pathogens into a capsule, and then at least they can't come out, but they also cannot be cleared. And these granuloma can be seen, for example, under the skin in this patient, but they're also in inner organs, and they can cause quite problems. And on the right side, you see an infection in a mouse with a fungus, Candida albicans, and this is something that usually only affects patients who are immunocompromised. So if you have a normally functioning immune system, these fungi are not a problem. And this oral infection in the mouse is called thrush. And this only happens usually in small children who have a inherited defect in the immune system. But if you come to the emergency room and you have this and you show this to the doctor and you are like 20 years or older, then the doctor knows, okay, it's something is seriously wrong with the immune system. Usually you have an infection with HIV, human immunodeficiency virus, that causes then other secondary infections because your immune system is not working. Yes, Alex.
At this point, I would like to add that the lack of nutrients also could cause the infection with Candida albicans, because this is something we also had in our, I know people who had that.
Yeah. So this is called a secondary immunodeficiency. Primary means, we will look at this also in more detail in a later video. Primary means something is wrong in your genes and you have had it from the beginning of your life. And secondary is something that you acquire during your life. And this can be either, the most frequent ones that we'll also talk in much more detail is HIV infection, but also lack of nutrients is a big problem, not only in certain patients, but also in poorer countries. Many small children are severely immunocompromised because they do not get enough food, and they have measles and candida and all these kinds of infections in a much worse way than would be the case here in Germany, for example. Okay, so if you remember the beginning, there were these big multicellular parasites that the immune system needs to fight, and they are very big. So they start from a few millimeters and can be 12 meters. So this is nothing that a single cell can take up or phagocytose anymore, and therefore we have a different system. And these are the mast cells and the eosinophils and the basophils, and they recognize certain structures of the parasite via antibodies. These are the red structures that act as adapter molecules, and then these cells will also degranulate, and they have a lot of toxic molecules in their inside. You can see this mast cell is full of these vesicles, and they contain so-called preformed mediators that are always ready. Basically, a mast cell is like a grenade, and once you pull the pin, it will just explode with this cocktail of toxic molecules. Histamine is an inflammation enzyme. Serotonin is also a molecule that regulates functions in your body, so more immune responses will going on. Then you have proteases that degrade, for example, proteins from these parasites, but they also degrade, for example, toxins from spiders, from snakes, or from scorpions. Very important. Then you have heparin. This is an anticoagulation drug, so it prevents the clotting of your blood, but it also prevents the normal function of these parasites. And then you have cytokines, messenger molecules of the immune system, that recruit even more cells of the immune system. As I said, this is preformed. This happens directly, and then after 2, 3, 4 hours, these cells produce also different mediators, because via the binding of the antibodies to the pathogen, an activating signal is transmitted into the nucleus, and then they produce certain molecules. They are called prostaglandins, thromboxanes, leukotrienes, and platelet-activated factors. These are all molecules that are generated from lipids, so fatty molecules, and they all induce very strong inflammation over very short distances in your body. So you will have a very strong inflammation reaction going on, and a lot of recruitment of other cells, and then together as a teamwork, these cells can then usually eliminate the pathogen. There are also more cytokines produced, chemokines. So cytokines tell the cells what to do, and chemokines, if you want to put it very, very simply, I know this is not correct, please don't bombard me in the comments, but chemokines just tell the immune cells where the infection is going on. So they mostly transmit an information about the location of the inflammation, if you want to put it very simply. Then growth factors are produced. This is a problem if the infection goes on for longer times. We see this also in allergic patients, where the tissue gets rebuilt due to these growth factors. Then we produce free radicals, and substance P is also something that is very toxic. The problem is this can also be triggered by pressure. So certain patients react with a pressure to the skin, with mast cell activation, or changes in temperature with mast cell activation, and then they have severe inflammatory reactions, and also allergens can do this. So far we have only looked at the mast cells, but there are also eosinophils and basophils. They all do similar things, so they degranulate in response to parasites. But what is different is the location. So we find mast cells everywhere in your body, directly under the potential points of contact with the outside. So we find them under the skin, under the epithelium, in the lung, in the gut, in the respiratory tract, it's the lung, or the reproductive tract. We find them also under all blood vessels, and they are ready to fight pathogens that invade your body. The eosinophils we find in your lung, in your gut, and your reproductive tract, and the basophils we find in your blood. And whereas you find like good amounts of mast cells always in your body, the number of basophils and eosinophils are usually quite low if you don't have an infection or an allergy. So they are secondarily upregulated if you have contact with pathogens. Therefore, they are also used very frequently in diagnosing different diseases. So if you have increased numbers of eosinophils or basophils, you might be allergic to something, or you might have a parasitic infection. So this system of mast cells, eosinophils, and basophils that fight these big multicellular parasites is very important. It usually works pretty good, not perfect, because these parasites, as you can imagine, are very complex beasts that also have developed many different ways to counterbalance the immune system. But it also can cause trouble. So if mast cells, basophils, and eosinophils are activated by actually harmless substances from our surrounding, this results in allergies. So here you see some examples, like eczema is an allergic inflammation of the skin, and this is what we talked about in the beginning. You see the skin is inflamed, people start scratching, and then also pathogens, bacteria, fungi get in there and cause even more trouble. Then on the right upper part, you have allergic conjunctivitis, so an inflammation of the eye. This is the most frequent allergic reaction in Northern Europe. Then you have milk allergy. After consuming milk, you don't feel so good. And on the right, you have a foot that was stung by a bee, and you see maybe that this is quite swollen, so there's also inflammation going on. Yeah, so this is the innate immune system, and then we will close out today's lecture with the adaptive immune system. So these are the cells that I talked about. I know I didn't talk about the NK and the NKT cells. They are a little bit more complicated. We will look at them in the next lectures in more detail. But we have also the adaptive immune system. These are the T and the B cells. And the adaptive immune system is then further divided into the cellular part. These are the T cells. And the humoral part, the B cells. So cellular, because it consists of cells. Yeah, this makes a little bit of sense. And there are different kinds of T cells. And humoral is a historical term. It describes the B cells and the antibodies that are produced by the B cells. And humoral means in the humerus. Humerus are your bodily fluids, and this is called that way because the antibodies produced by B cells can be very easily and in very large amounts be isolated from your bodily fluids, like your blood, for example. Cells of the adaptive immune system, the T and the B cells, are called lymphocytes. That's the overall term for them. And they arise from the common lymphoid progenitor. We have seen this cell before. This is the cell in the bone marrow that has decided to become cells of the immune system. And this can develop then into either a naive T or B cell. You can see here a difference, Alex would say, because one is green and one is blue. But if you just look at them under the microscope, you wouldn't be able to tell a difference because they look very similar. But once they get into contact with a pathogen, they will differentiate and they will proliferate. What does this mean? Differentiate means they turn into something else. They look different because they transition from the naive fight-ready state into a I go to war state, I would call it like this. And proliferation means they form millions of identical daughter cells that fight then the pathogen. This is what we call an activated state. And once they reach the pathogen, they will produce antibodies in the case of the B cell, or secrete cytokines, for example, in the case of the T cell. So this is then the effector cell and the effector function. So this is how they actually kill the pathogen. There's a quite interesting difference between how cells of the adaptive and the innate immune system recognize pathogens. So cells of the innate immune system use these pattern recognition receptors that we talked about. And here's a dendritic cell in the middle that needs to initiate immune responses against viruses, parasites, fungi, and all other kinds of pathogens. And it does so by using a limited amount of these pattern recognition receptors. There are maybe 3-4 dozen of these pattern recognition receptors that are used to recognize these pathogens as dangerous and foreign. But on the side of the adaptive immune system, we have, for example, 6 different T cells, and they have very strictly tailored, very specific T cell receptors. These are the structures on the surface of the cell that they use to specifically recognize one specific pathogen. So the red cell recognizes the red virus and nothing else. The green cell is recognizing a green virus. The blue cell is recognizing the blue parasite, and so on. So they have very tailored, very specific receptors, whereas the innate immune system has to cope with 3-4 dozen of receptors that are the same in all of us. So Alex has the same set of pattern recognition receptors than me and Hannah, but our adaptive immune system will look different because this is as we will see, generated randomly. And the T cells that are flowing around in my body and in Alex's body are different because we have gone through different infections. So if I have had a flu like last week, I would have a lot of flu-specific T cells, and if Alex would have had, I don't know, a bacterial infection, she would have a lot of antibacterial T cells still around. So the difference is that the cells or the receptors of the innate immune system are germline encoded, so they are hard coded in your genes. They are all the same in all of us because we all have the same genome, more or less. But the receptors of the adaptive immune system are generated by something that we call somatic rearrangement. Fancy term, I know. You probably don't know what this means. Therefore, I will explain it very briefly. We will take much more time. I know how much time we take because we've already recorded that lecture. It takes like 30 minutes at least to understand it. But the lecture's there. We will load it up later and then you can see it. So if you look at a T cell, it has a T cell receptor on its surface. That is the green structure. And this is encoded by a gene in the nucleus of this T cell. And what a very famous scientist, Dr. Tonegawa, found out in the 80s is that the T cell receptor is not encoded by a single gene, but it's encoded by a set of different fragments that are randomly combined with each other. You can imagine this like Lego bricks, and you take one red brick, one green brick, and one yellow brick, and you put them together and you get a T cell receptor. And if you take a different red brick and a different green brick and a different yellow brick, you get a different T cell receptor. So there's combination going on in how these receptors are generated. It's even a little bit more complicated. You see here the structure on the genomic level. So the T cell receptor consists of two proteins. One is called alpha chain and the other one is called beta chain. If you look at the fragments for the alpha chain, this is called the alpha locus. They are different segments that are called V alpha and J alpha, and there are like 70 to 80 different V segments and 61 J segments. V stands for variable and J stands for joining. And then one is randomly picked. In my example here, it's the first J segment, the first yellow segment, and the third V alpha segment. And they are combined in a process that we call recombination, and all the sequence information between after the third V alpha segment and the first J segment is deleted, is excised as a plasmid from the DNA of that cell, and it gets lost during subsequent cell division. So this is a loss of genetic information. These cells, once they have put together a receptor, do not have the complete genome anymore. This is something that is very curious. And this gets then further transcribed, spliced, and translated into a protein that then ends up on the surface of the cell. If you look at the receptor, you see it has a blue part that is anchored into the wall of the cell, and this is encoded by the constant segment, the C alpha. This is the same for all T cell receptor because this is the structural framework it needs to correctly fold itself and to integrate into the membrane. But the tip of the receptor, where it recognizes the pathogen-derived peptides presented via MHC molecules, is made up from these fragments. And there, basically all kinds of combinations of fragments can be used to generate different receptors. So that would be 80 times 61. I don't know how much this is, but it's a few hundred, probably a few thousand. But additionally, in a process that we will explain in much more detail, there are also random sequences inserted in the region between these two segments that result in frame shifts and additional amino acids. So it's basically an endless amount of sequence information that can be generated, and therefore we can generate up to 100 million different T cell receptors, also against structures that we have never seen. For example, I always choose the example of the COVID-19 pandemic. So COVID-19 didn't exist before 2019, but still our immune system has managed to form T and B cell receptors against COVID proteins. For the second chain of the T cell receptor, it's the same process, but a little bit more complicated because we have two additional segments, the D beta 1 and D beta 2 diversity segments, but you see also they end up at the tip of the T cell receptor beta chain that then binds the peptides. We distinguish three classes of T cells: the T helper cells, the cytotoxic T cells, and the regulatory T cells. We have learned a lot about the T helper cells already. They are called CD4 positive T helper cell. This is just an additional receptor on the surface of the cells that can be used to easily identify them. And their main functions are producing cytokines as managers of the immune system to recruit and activate different cells of the immune system to the place of infection. This is something that we have talked about extensively already. Then we have the CD8 positive cytotoxic T cells. They are called the killer cells also. Strong words. Cytotoxic means cyto is again the cell, and toxic is like toxic or killing. So they can kill cells. And once they are formed in the thymus, they are released into your body, and they constantly scan MHC molecules. They are different MHC molecules. If you look very closely, they're called MHC class 1 molecules on the surface of all your cells in your body that have a nucleus. So all your cells in your body that have a nucleus have these MHC class 1 molecules, and they present peptides from the molecules that they work on their inside constantly. And if the cell gets infected by a virus, it stops producing, I don't know if this is a liver cell, liver peptides, but it starts producing virus particles, and therefore we will have virus peptides on the surface of these MHC molecules. And then the cell gets activated because it knows, this turquoise peptide is from a virus. I should kill that cell before it produces more virus proteins. And then it releases two molecules. One is called perforin, the other one is granzyme, and the perforin punches holes into the cell wall of that cell, and the granzyme then goes to the nucleus and it induces apoptosis. And apoptosis is something that we call programmed cell death. So the cell will basically digest itself and die before it can produce more viral molecules. And you can see this is a very specific process because only the liver cell in the middle is killed, but not the one on top and not the one on bottom that are not infected because they do not show virus peptides via the MHC molecules. So the cytotoxic T cells are the ones that do the actual work. They kill infected cells. And then we have the regulatory T cells. So imagine this is an antigen-presenting cell that has a very bad day and tries to activate a T cell that actually recognizes a part of my brain. And if the cell would be activated, it would destroy my brain and I would probably die. So that would be pretty bad. I would become autoimmune. And there are cells that prevent this, and they are called regulatory T cells. We will explain in much more detail why they are there and how they are there and how they work in a later lecture. But basically, they recognize the same self peptides, so peptides generated from our own proteins that are actually not dangerous. And they then can shut down the antigen-presenting cell by not using co-stimulatory molecules, but using co-inhibitory molecules. That's the yellow one. So it basically tells the antigen-presenting cells, stop what you're doing, you're having a bad day, go to bed, rest a little bit, but don't start any more immune responses, please. And it can also shut down the possibly autoimmune T cell by secreting the red guys and girls. These are the suppressive cytokines like interleukin 10 or transforming growth factor beta. They basically prevent the activation of that cell. This is good. This is a safety mechanism, so then it doesn't do anything anymore. This is also done to the antigen-presenting cell, so it's basically my sentence with go to bed and sleep and try again tomorrow. And they also produce perforin and granzyme that we know from the CD8 positive T cells. And this is a more extreme measure of protection because they then kill the cell and then the autoreactive cell is gone. There's no try again tomorrow for that cell if it is that dangerous. So therefore, these regulatory T cells, which are a subset of the CD4 positive T cells, can then suppress the activation of the other T cells. As I said in my disclaimer, it's much more complicated. So here you see the different factors and the different cytokines and the different transcription factors that are relevant for the different T cells. And you see that also there are different kind of CD4 positive T helper cells, not only the regulatory T cells, but we have, for example, Th1 cells, Th2 cells. The Th1 cells are like the antibacterial ones. The Th2 cells fight these parasites. It's very complicated, I would say. And if you need to understand this, you're probably doing a PhD in immunology. So Alex and Hannah understand this, but you probably don't at this point. I just wanted to add the information that you have seen that it's much more complicated. So there are basically certain combinations of cytokines, that is what is shown on top, that are produced by the antigen-presenting cell. They result in the activation of certain master transcription factors in these cells, and they then turn on or off quite a bit of genes, and then they turn into these different cells. And this is why signal 3 is so important, because this then triggers this cascade, and then the cell knows exactly, there's a blue bacterium, I need to turn into a Th1 cell and fight that bacterium. So also the cytokines produced by the CD4 positive T helper cells activate the cytotoxic T cells. These are the red guys and girls, and they activate B cells. And the B cells are like the last cells we're going to look at today. The B cells are the main antibody, or the only antibody producers in our body. They can also take up antigens from pathogens, for example, like the red one, via their B cell receptor, BCR. Spoiler, this is a membrane-bound antibody molecule. This binding to the B cell receptor results in the antigen being taken up into the inside. These are usually protein or protein structures, or carbohydrate structures or sugar structures. They get then degraded inside the B cell into peptides, the proteins that are then presented via MHC molecules. So it's the B cell is an antigen-presenting cell, and this uptake, processing, and in last consequence, the presentation via MHC class 2 is signal 1 in the B cell. And they present the antigens they have found to the T cell. And the T cell now acts as an authorizing cell to activate the B cell, because this T cell, you have to imagine, was shown the same peptide by a antigen-presenting cell, like a dendritic cell, in the lymph node 10 minutes before. And the dendritic cell basically said, if you see this pink peptide, it means that there is a pathogen around, and I allow you to make an immune response. So therefore, this naive T cell was shown this peptide. It turned into an effector cell, and then it looks around in the lymph node if it finds a B cell that shows the same peptide. So this is how it makes sure that only an immune response is initiated against the pink pathogen. Then it gets co-stimulatory signals via CD40 ligand and CD40. This is signal 2. And now it's the other way around. Now the T cell activates the B cell. In the T cell activation, it was the T cell that was activated by the dendritic cell via the co-stimulatory molecules. And also the T cell now produces cytokines, the signal 3, that tell the B cell what kind of B cell it should become, like an antibacterial, antiviral, or an antiparasitic, or antifungal B cell. So once the B cell gets activated, it turns into an antibody secreting plasma cell. Antibodies are also called immunoglobulins, Ig. And these cells are quite big and quite active. They can produce 10,000s of antibody molecules per second if they are fully activated and fully fed with nutrients. So antibodies have 3 main functions, and that the 2 of them do not fall asleep. I asked them what are the 3 main functions of antibodies. 1 we have seen already, maybe 2 we have seen already. Yes, Hannah?
The one we have seen already was the complement activation.
Yes.
If I remember correctly.
Mm-hmm.
And then, should I also do the other?
Yes.
It's also neutralization and opsonization.
Perfect. So we have neutralization. This means that antibodies bind to the surface of pathogens like this virus here, and they just put them into big aggregates, lump them together, and then they can't swim freely. And for example, viruses use certain fusion proteins on their surface to fuse with their target cells. And if these fusion proteins are blocked by antibodies, the infection gets blocked, and therefore these viruses are a lot less harmful. This is, for example, what we wanted to have during the COVID-19 pandemic. We wanted to induce neutralizing antibodies with the vaccines that block the virus from infecting our cells. Then we have the opsonization here. The antibodies act as adapter molecules to mark pathogens as foreign and dangerous and allow cells, even if they do not have the pattern recognition receptors, to recognize them as foreign, to recognize them as dangerous. Here the antibody acts as an adapter molecule. So we've seen this, for example, with the antibodies that bind to the mast cells and then bind to the pathogen. Here the antibody acts as an adapter molecule. And the term opsonization means marking as foreign by the addition of certain molecules. And then we have the activation of the complement system that is triggered, at least in the classical pathway, by the binding of the antibodies. So until now, I hope the antibody always looked like this, but actually there are different kinds of antibodies that we call isotypes, and there are 5 different kinds of antibodies. And to make it not too complicated, but I already called it this like before, and I was stopped by Alex. So I was instructed to say that the first antibody that B cells made is called IgM. And these antibodies are okay at dealing with most infections, but not very good, not very efficient. We will look at this in also a next slide. But then during an infection, the cell can also decide to make other types of antibodies. Here the IgG, the IgA, and the IgE are the most frequent ones. The IgG has not so many functions in the activated B cells, probably only during a B cell development. But they have then different functions. And you see in my example, the part of the antibody that binds the antigen, the upper red part is always the same. What gets exchanged is the lower part and only the heavy chain, if you know what this means. And therefore these antibodies have different effector functions. Antibodies can also be monomeric, so it's just one antibody molecules. They can be dimeric, so there are 2 antibodies, 2 identical antibodies combined via a joining chain. This is, for example, in IgA the case. And you can have pentamers, where 5 antibodies, 5 identical antibodies are combined into one big antigen-binding conglomerate. And this is what usually the B cell starts with, because I said the IgM is the first antibody that is produced. It's not so great, but it works somehow. And this lack of binding quality is compensated by quantity. So a normal antibody, a monomeric one, has only 2 antigen binding sites. A dimeric one has 4 antigen binding sites, and a pentamer has already 10 antigen binding sites. And therefore you get a compensation for lack of quality by adding of quantity. So if you look at the functions of these different isotypes, you see neutralization. And if you look at which isotypes can do good neutralization, it's the IgG. The IgA is already a bit worse, and the IgM is not so good at neutralizing. Then we have opsonization. There the IgG is good, and the IgA and the IgM are not good. And for the complement activation, it's again the IgM and the IgG and the IgA. And for the mast cell activation, it's only the IgE. So, yeah, a lot of information, complicated. You might ask yourself, as Alex is already doing, why this is the case. And I can tell you, if you think about it, it makes a little bit of sense. Yes, Hannah?
Sorry, I did not want to interrupt.
No.
But my question when I see the picture for the opsonization is, do the antibodies first bind to the pathogen, or do they bind to the receptors on the other immune cells?
Excellent question. It's a bit complicated. So if you look at the right upper part that are called opsonization, there the antibody first binds to the pathogen and then to the immune cell. And for the mast cell activation, which is also opsonization, because also here the IgE acts as an adapter molecule, the antibody first binds to the mast cell gets armed with the IgE, and then it binds to the pathogen. That's why I also didn't call this opsonization. It's quite complicated. In the end, the functional consequence is the same. You get the antibody working as adapter molecule and immune cell activation, but it has vastly different consequences for treatment of diseases caused by these antibodies, because for the IgG, IgA, and IgM dependent diseases, you can just filter out the antibodies. For the IgE mediated diseases, you somehow have to get rid of, or you have to dissolve the antibody from the surface of the mast cell, which is much more difficult.
Okay, so both both exist, and it depends on the type of antibody, and it depends then or results in different diseases like allergies with IgE antibodies binding to the receptors of the or basically the receptors of the mast cells binding to the antibodies first, or with other diseases where for example, IgM antibody binds to the pathogen first.
Yes, exactly. And if we now look at the differences, they make a little bit of sense if you think about it, because the IgA we find in the mucus, so outside of your body. There are no immune cells outside of your body, hopefully. Otherwise you would bleed quite profusely. And therefore it doesn't make sense to opsonize, because for opsonization you need immune cells to bind to the antibodies, and there are no immune cells outside of your body. Neutralization makes a lot of sense, because you want to prevent by IgA binding the invasion of the pathogen into your body. And also complement activation by IgA makes no sense, because there is no complement proteins outside of your body. Therefore, it doesn't make sense. The IgG is very good at everything. So this is just an all-rounder. And the IgM is very good at complement activation, because it's the first antibody that is made during infection, and therefore maybe not so many immune cells are around during that time of infection, but all the complement proteins are still around. Yes, Alex, the B-cell expert is speaking.
For the complement activation, on the picture is only one antibody depicted, but you said that IgM is mostly pentameric.
Yes.
Is the pentamer activating the complement system,
yes.
or is it then a monomer of IgM?
Yeah, no, it's a pentamer, and there are also two different conformations. There's one that looks like a hand like this, and there's one that also flat, and also the one that looks like this then binds nicely to the surface and triggers the complement cascade quite proficiently. Yeah.
Thank you.
Okay, and also neutralization, does not make so much sense for IgM, because there's not much cells around, and also opsonization doesn't make so much sense, because at the early stages of infection there are just not so many cells around. This is then reserved for the IgG antibodies. So altogether, this cascade of initial activation of antigen-presenting cell, promotion of T cell activation, and then phagocytes, inflammation, CD8, T cell activation, B cell activation, usually results in a very efficient elimination of the pathogens by these different methods. There's one more thing we need to look at, and this is the generation of memory, because if it would be like this, every infection would lead to the same reaction, and you would get as sick from the first infection as you would get from the second infection with the same pathogen. But as you maybe know, once you have a pathogen for the first time, you might be very sick, and the second time you are much less sick, or probably even don't realize that you have the disease anymore. And this is because we generate memory cells. So imagine you have been infected with a fungus, this violet fungus. It activates antigen-presenting cell that activates a fungus-specific T cell. This expands into millions of identical effector T cells that fight the fungus, and then the number of fungi in your body will hopefully decrease to a very low level. So there are very few fungi or even no fungi around, and therefore no more fungi are taken up by antigen-presenting cells, no more T cells are activated, and therefore the T cells will die at some point. This is what we call the 3 steps of an immune response. You have the initiation, where the pathogen gets sorted out as dangerous and foreign. An immune response is initiated. Then you have the expansion, where you generate millions of identical cells that fight the pathogen, and at the end, these cells have to contract because they need to make space for example, the next viral infection. But some of them will turn into memory cells. So these are usually ones that are very good at eliminating that fungus. And they are then kept alive for decades in your bone marrow by certain cytokines like IL-7 and IL-15. And they can then be reactivated, taken out of cold storage, if you want to call it like this, once you have again contact with the fungus. And then it can efficiently eliminate that fungus. And so efficiently, it basically starts where the other cells left off at the end of the infection. And this results in this very efficient elimination of the pathogen. Therefore, we distinguish between primary and secondary immune responses. So primary is when you have first contact with the pathogen, then all the things I explained to you today take place. You eliminate the pathogen. It takes a little bit of time. If you have the first antibodies, like 4 or 5 days, then you have the IgGs, it's like 7, 8, 10 days. And then you eliminate the pathogen. And if you are then again confronted with the same pathogen, the same antigen, that's the blue one, you make the secondary immune response, which is much higher, much faster, much longer lasting. But if you are in contact with a different antigen, antigen B, that's the orange one, you make again a primary immune response. So this is very elegant and allows us to live a life where we don't have infections all the time. And if you have small children, they do not have formed these memory responses yet, and they are sick very often. So 12 good infections per year are normal for small children, because they first need to build up this memory repertoire. So for them, everything is basically a primary immune response. So if we sum up, we have the innate and the adaptive immune system, and the innate immune system is immediately available directly after birth, more or less. They react within minutes and hours, so they eat up pathogens within seconds. The mast cell activation works within minutes, but they have to rely on a fixed set of pathogen recognition receptors that is the same in all of us. It's quite effective, but it does not form memory, as I told you. We will spend a whole lecture where we push this around and explain you that they can form from memory, but I think at the moment on your level, you can just save it as there is no memory formed. The adaptive immune system is only available after some months, because it takes a little bit for the immune system to start and to make your first T and B cell responses. Also, once you initiate T and B cell responses, it takes some days and weeks, because first you have the antigen-presenting cell that gets activated. It needs to find a specific T cell that needs to be activated, proliferate, differentiate, find a B cell that needs to be activated and produce antibodies, and so on. So it takes some days and weeks, but they rely on these randomly generated T and B cell receptors and can, in theory, recognize an unlimited amount of different structures. So it's much more effective and much more tailored and much more specific. And they develop immunological memory. And if we put the 4 barriers I told you today, again, here on a last slide, you see we have the anatomical barriers that keep out the pathogens. Then we have the complement system and the antimicrobial peptides that directly start killing the pathogens once they are inside the sterile parts of your body. Then we have the innate immune system and we have the adaptive immune system. And every barrier kills a lot of pathogens. And the ones that are left, for example, that have made it beyond the anatomical barriers, are killed by the complement system, and the ones that are not killed by the complement system are then attacked by the innate immune system, and the few that are left can then be eliminated by the adaptive immune system. So this is a mesh of processes that are connected to each other, and in most of us, it is enough to eliminate most of or all of the pathogens that we are confronted with in a daily basis. So with this, we are at the end of today's lecture. I hope we could bring a little bit of clarity into the complex immune system. We know this is complex, and we used as little as possible fancy terms and complex things. We try to make it always as easy as possible. If the two of them have no further questions, then I thank again the two of them, because without their help I couldn't do them. Without their questions, the videos wouldn't be as good as they are, I say. And it is really appreciated that they take their time. If you, dear viewer, have additional questions, just put them in the comment section. We try to get back as fast as we can. And with this, I would leave you. Maybe we see us again in the next video, where we will talk about the innate immune system in much more detail. See you.
Bye.
Great. Then we press stop, or?
Could you hear my belly? It was so loud.
No.
No.
Oh, God.
Ready?
No, but just do it.
Just do it, okay. Feels like we did this yesterday. But yeah.
But it's 1 week ago.
That's 1 week ago, yeah. And it's again 7:50 in the morning. Oh, it's perfect. There's an airplane. Sorry, I have to wait for this one.
I don't understand what you say.
There's an
Me
airplane.
neither. But it says silent, so I think it's okay.
But it sounds like he would pray. What he's praying to? Is there a god of science? Is this?
Shut up.
Okay, no. There was an airplane, and I need an airplane for correction purposes. That was the perfect timing. Stupid airplanes. Otherwise, I have to hunt through the 2-hour file to find an airplane. No, it was directly at the beginning.
Which shouldn't be as difficult.
Yeah, but you need to find one where I don't talk. Yeah, that's the problem. I talk a lot here. Alex?
Yeah.
Yeah? No. No.
Nah.
Okay, then. Clap from Alex. No. But it's okay. Just do one straight from me. Thank you. And Hannah. Yeah. Hannah, I hear. Okay.
Tell me what I'm doing wrong and I will change.
I think I need to send you to a clapping university.
Clapping University.
Yeah.
Okay.
So it's theater, 1 course, 1 semester, 12 hours. Different forms of clapping.
Okay.
So, we do overview over the immune system.
How long will this take?
As long as it
1.5
takes, I don't
hours
know.
if he's
If
walking through.
But we need to ask questions for
Yes.
the listeners, so it will take probably 2 hours.
Probably. So, do you have other appointments, or what? Do you have more important
Let's
things to do?
begin.
Yeah.
I need to work in the afternoon.
Oh, me too.
Me too.
So,
But
The only difference is Stefan is on vacation today.
yeah.
little bit jealous.
Yeah, but still I do video editing all day long. Probably this is done by so-called
Tight junction.
times. It's not my week, Alex. It's not my week.
I don't want to
I
correct
just
the
cut it out.
professor.
I just cut the whole pollen receptor thing out.
Not yet
Or-
professor, but maybe.
Never
soon.
will be professor. I think you will have your PhD before I have my professor. Easily. As I said, I have a disclaimer. Everything's fine. Okay. I will try my very best.
I know. You will manage.
Yeah.
We will manage.
Yeah. Do you want? Yeah. That would be much easier. Basically, if you have an infection, you make an antibody that is good to neutralize the basically, if
Anything? Just the picture before. I don't think that it's then clear that IgM is the first one.
Yeah, you are, of course.
Because it looks like IgE.
Actually, yes. And the sentence was like, no, it's differentiating. The first antibody is differentiating into IgM. No.
I do it again.
To take care that people don't think that antibodies differentiate into IgM.
This is when you give a lecture with 2 B-cell experts. I'm just a supervisor, I have no clue. Yeah.